Modification of myocardial autorhythmic cells. Here's the graph that we've talked about for myocardial autorhythmic cells, which do not exhibit a resting membrane potential. Rather, they have this pacemaker potential that leads to an action potential where we have a depolarization event, repolarization event, another pacemaker potential, depolarization, repolarization, and that continues on and on and on, really until the end of life or until now. This can be modified. So keep in mind, I've suggested that these myocardial autorhythmic cells are going to depolarize and repolarize roughly 80 times per minute. Resting heart rate, depending on an individual, it is certainly highly variable, but let's just suggest it's 60 beats per minute, which would correlate to the amount of times that the MAC is firing. And that diminished heart rate is the result of parasympathetic activity, which is going to increase the permeability of potassium and decrease the permeability of calcium, specifically in the area of the resting membrane potential. This graph right here. And as we can see, we are starting lower than -60 because the increased permeability of potassium is increasing the efflux of potassium. That is a positive charge leaving the cell, making the cell more negative. And also the decrease in calcium in this region is causing this slope of the pacemaker potential to not be as steep as it normally is. So it takes longer to reach threshold. So there's two factors here with parasympathetic activity, which is what we're talking about here. Parasympathetic activity is going to decrease the rate of myocardial autorhythmic cells due to the release of acetylcholine binding to muscarinic receptors on autorhythmic cells. We can also get sympathetic activity influencing MACs, and that's the release of norepinephrine and epinephrine from the adrenal medulla. That is a neurotransmitter and hormone respectively, that are going to increase the rate of myocardial autorhythmic cells due to sympathetic activity. And this is a result of increased calcium permeability and increased sodium permeability. So we get a much steeper pacemaker potential due to the increase in sodium permeability and the increase of calcium permeability, and super steep right here in the initial depolarization of the action potential due to that increase in calcium permeability. We get the repolarization event, another steep depolarization event due to the increase in sodium permeability and calcium permeability. That is sympathetic activity, which is norepinephrine and epinephrine binding to beta-1 receptors of the autorhythmic cells.